Good morning, my dear students. Welcome to our Zoology online class. Hope you are very happy. And dear students, in the last class we completed the fourth chapter. Okay. So in the last class we studied about the chromosomal abnormalities. Okay, chromosomal disorders or chromosomal abnormalities. And the reason for this chromosomal abnormality is changes in the number or changes in the structure of chromosome. So if any errors takes place in the structure of chromosome or if any errors occur in the number of chromosome, it leads to some disorders. That disorders are called chromosomal disorders. Okay, and four types of two autosomal disorders and two allosomal disorders we have studied in the last class. Okay, for example, the Down syndrome, Patur syndrome, and then Klinefelter syndrome, Turner syndrome, like the four chromosomal disorders we have completed in the last class. Okay, so hope you all studied these question answers. It's togetherly very important. Chromosomal disorder togetherly very important for 5-mark questions, and each disorder may be separately may be asked for 2-mark question. Okay, and some 1-mark questions also there. So I hope you all studied this chromosomal disorder. Okay, my dear students, today we are going to start a new lesson. Okay, so today we are going to start the 5th lesson. So our lesson's name is molecular genetics. Molecular genetics. So it's a very big lesson. So in this lesson we are going to study about some important topics. Okay, topics means the properties of gene, structure of DNA, then transcription, translation, okay, the process of transcription, process of translation, then genetic code, DNA fingerprinting techniques, okay, like the different topics we are going to study from this lesson. Okay, and this lesson also very important for 5-mark question, 5-mark and 3-mark question. So dear students, listen very carefully. Now we are going to start the 5th lesson, molecular genetics. Okay, say genes. The last chapter also we studied about these genes. So genes are the physical and functional unit for hereditary of hereditary. Hereditary meaning transformation from one generation to next generation is called hereditary. Okay, so genes are the physical and functional unit of the hereditary process. Okay, and because of the presence of the genes only, some characters are transferred from one generation to next generation. Understand a few? So that only the genes are otherwise called the physical and functional unit of hereditary. Understand a few? Then, see, first the concept of gene was first explained by one famous scientist. His name is Johann Gregor Mendel. Okay, Gregor Mendel. He only first explained the concept, okay, or the basic concept of the genes in the year of 1860s. So during 1860s, Mendel only first explained the basic concept or the concept of the genes. Okay, and when he was doing many experiments, he isolated the genes, but he didn't give any name to these genes, just he called it as a factor. Okay, so he Mendel only first explained about these genes, but he didn't give this name gene, but instead of this word gene he used the word factor. Okay, he used the word factor. Understand a few? Then in the year of 1909, another one scientist, his name is Wilhelm Jorgensen. Okay, so this person only first put or given the name gene. Okay, so Gregor Mendel used the word or called factor. Then another person, Wilhelm Jorgensen, only first replaced this word by using the word genes. Okay, by using the word genes. Understand a few? Then in the year of 1902, okay, in the year of 1902, another one person, his name is Sutton, S-U-T-T-O-N, Sutton. Okay, this person only first explained the classical concept of the genes. First explain the classical concept of the genes. According to this classical concept, or according to the view of Sutton, genes playing a very important role in the hereditary process. in the hereditary process or inheritance process. And each and every gene is occupying a particular location in the chromosome, in a chromosome. Okay, so the location of the gene is chromosome confirmed. Okay, so in the classical concept explained by Sutton says the gene is playing a very important role in the inheritance, and each and every gene is occupying a particular position in a chromosome. Understand of you? So dear students, now we have studied 3 important 1-mark questions. The first question is, who first explain about this gene? Okay, Johann Gregor Mendel. And the second one, who given or describe the word gene? Okay, Wilhelm Johannes. Okay, then the classical concept of the gene was first explained by who? Sutton. So now we completed 3 1-mark questions. So it's a small information about the gene. And dear students, now we are going to study about the first 5-mark question of this chapter, properties of genes. Okay, so the first 5-mark question is properties of genes. So in your book there are 6 properties are explained. Okay, 6 properties of a gene is explained in your book. So the first property is number of genes. Listen very carefully. Here number of gene means in a particular organism's body cell, always the number of gene is higher than the number of chromosomes. Okay, you all know in our body cells only 46 chromosomes only they are. But each and every chromosomes consists of large number of genes. So the number of genes is always higher than the number of chromosomes. So this is the first properties of the gene. Second property, arrangement. Each and every genes are arranged in a linear structure, like the beards on a string. Okay, so the arrangement of the gene is linear arrangement. So this is the second property of a gene. Then third one is locus. Locus meaning here location. Already I told you, each and every genes are present only inside the chromosome. Okay, so every genes are occupying a particular location or particular position in a chromosome. So this is one of the important property of gene. So this is the third property. The fourth one is alleles. The next property of the gene is alleles. Alleles means the genes are always expressing in a alternative form. So this alternative form of a gene is called allele. The alternative forms of the gene is called allele. Then the fifth property is mutation. Sometimes some genes can involve in sudden change. Okay, so this is called gene mutation. This is called gene mutation. So if any genes involved in mutation, it leads to some changes in their structure or position or in their compositions. Okay, so when a gene is involved in mutation, some changes takes place in the structure or in the position of the gene. So that is called mutation. So mutation means sudden change. The next one is self-duplication. Sometimes a gene, a single gene can take its own copy, okay, own additional copy. So this nature or this property is called self-duplication. That's all, my dear students. So first 5-mark question of this chapter is properties of genes. So there are 6 properties are explained in your book. So the first property is number of genes. Second property is arrangement. Third one is locus. Fourth one is allele. Fourth one is mutation. And the last one is self-duplication. Hope you understood this 5-mark question. Okay. Then now we are going to study about 2 2-mark questions. The first question is one gene, one enzyme hypothesis. One gene, one enzyme hypothesis. Here two scientists. One scientist name is called John Baden and second scientist name is called Edward Tata. So these two people togetherly did one experiment in a mold or in a fungus. Name of this fungus is called Neurospora crassa. So in the body of Neurospora crassa, these two scientists conducted one experiment. And according to this experiment, one gene only controlling the production of a single enzyme. So enzyme's production is controlled by a single gene. So this hypothesis is called one gene, one enzyme hypothesis. Hope you understood this first 2-mark question. The second 2-mark question is one gene, one polypeptide chain hypothesis. One gene, one polypeptide chain hypothesis. Here, each and every enzyme molecules are made up of a particular number of polypeptide chains. Some enzyme molecules may be made up of 2 or 4 polypeptide chains. Some enzyme molecules may be made up of more than 10 polypeptide chains. Okay, so like this, each and every enzymes are made up of some polypeptide chain. Here, one gene, one polypeptide chain hypothesis, meaning the production of a single polypeptide chain is controlled by a single gene. So one gene is controlling the production of a single polypeptide chain. This hypothesis is called one gene, one polypeptide chain hypothesis. Here, one gene controlling the production of enzyme is called one gene, one enzyme hypothesis. Hope you are understood the difference between these two mark questions. Understand, my dear students? Understand? So, hope you are understood one 5-mark question and two mark question. Then, next topic is in search of genetic material. The next topic of this chapter is in search of genetic material. See, many experiments was conducted by many scientists to prove the genetic material of the living organisms. Some scientists says DNA is the genetic material. Some scientists says the RNA is the genetic material. Some scientists saying the protein molecules are the genetic materials. Okay, so in this topic we are going to study about that the DNA is the genetic material. So listen very carefully. See, in the year of 1848, one scientist, his name is Wilhelm Hofmeister. Okay, Wilhelm Hofmeister. Okay, he observed one important one important factor in the cells. That means the nuclei of the cell sometimes changed into a rod-like structure during the mitosis cell division, and now this structure is called chromosome. This structure is called a chromosome. So, in the year of 1848, Wilhelm Hofmeister observed that the nuclei of the cell is converted or changed into a rod-like structure during the mitosis cell division, and this structure is called chromosome. This structure is called a chromosome. Then, next point. In the year of 1869, another good scientist, his name is Friedrich Meisker. Okay, Friedrich Meisker. He observed that the nuclei only converted into nuclein. Okay, so he called the nuclei as nuclein. He called the nuclei as nuclein. Then, next, in the year of 1889, another good scientist, his name is Altman. Okay, so he said, he called this nuclein as nucleic acid. He called this nuclein as nucleic acid. Hope you understood these points, dear students. See, in the year of 1848, Wilhelm Hofmeister said or observed that the nuclei of the cell is inverted or converted or changed into a rod-like structure. This rod-like structure is called chromosome. Then in the year of 1869, Friedrich Meisker, he said he called the nuclei of the cell as nuclein. Then in the year of 1889, Altman called the nuclein as nucleic acid. Understand, dear students? Then another one scientist, he is a very famous scientist, Griffith. Okay, this person, Griffith, used the bacterial species to conduct one experiment. Name of his experiment is called bacterial transformation experiment. Name of his experiment is called bacterial transformation experiment. Okay, this experiment is the first proof that the DNA is the genetic material in some organism. Okay, so the Griffith experiment, name of the experiment is called bacterial transformation experiment. So this bacterial transformation experiment of Griffith is the first proof that the DNA is the genetic material. Okay, but here one drawback is there. He didn't understand. Griffith don't know the reasons for this bacterial transformation. Okay, he didn't understand the causes of this bacterial transformation. Hope you understood this point. So, some other scientists in the year of 1944, Avery, McLeod, McCarthy, these three people, togetherly repeated the same bacterial transformation experiment of Griffith. Already Griffith done this bacterial transformation experiment. Okay, so here he didn't understand the causes or the reason for the bacterial transformation. So these three people again repeated the same experiment, okay, to prove that the DNA is the genetic material. Okay, listen very carefully. See, according to these people's experiments, they observed a DNA, RNA, and proteins from one type of heat-killed bacteria. Name of the bacteria is called S-strain bacteria. Listen very carefully, dear students. So these people isolated the DNA, RNA, and proteins of S-strain bacteria. And this S-strain bacteria's DNA, RNA and protein is now introduced into another one bacteria. Name of the bacteria is called R-strain bacteria. Name of the bacteria is called R-strain bacteria. When this substance of this S-strain bacteria is added into the body of the R-strain bacteria, they observe some changes in the R-strain bacteria. Okay, actually, the outer surface of this R-strain bacteria is rough in nature. Okay, rough in nature. But when they add these DNA, RNA, proteins of this S-strain bacteria into the R-strain bacteria, the rough outer surface of this R-strain bacteria is converted into smooth nature, smooth surface. Okay, and now this R-strain bacteria is the pathogenic bacteria. So before adding the substance of this S-strain bacteria into the R-strain bacteria, the R-strain bacteria is not a pathogenic bacteria. After adding the substance of this S-strain bacteria into the R-strain bacteria, this R-strain bacteria is converted into a pathogenic bacteria. Pathogenic meaning disease causing or dangerous bacteria. Understand up you? So 2 points I said here. Okay, so 2 changes we understood in the body of the R-strain bacteria. What are the 2 changes? One is the rough outer surface of this R-strain bacteria is converted into or changed into smooth nature, and this non-pathogenic R-strain bacteria is converted into pathogenic bacteria after adding the DNA, RNA and proteins of this S-strain bacteria. Hope you understood, my dear students. Then, now they added one enzyme in the R-strain bacteria. Name of the enzyme is called DNase. Okay, so when they added this DNase enzyme in the R-strain bacteria, immediately the transformation capacity is lost. Okay, so the R-strain bacteria lost their transformation capacity. Okay, and they added another 2 enzymes, RNase and protease. RNase and protease. But this RNase and protease not affected the transformation capacity of the bacteria. So when they add the DNase enzyme in this R-strain bacteria, the R-strain bacteria's transformation capacity is lost. When they add this RNase and protease enzyme in the bacteria's body, this is not affected the transformation capacity of the bacteria. Understand, my dear students? So this is one of the very important proof for the DNA is that the DNA is the genetic material in the bacterial cells. Okay, then this experiment says that the DNA of one type of bacteria, just imagine S-strain. Okay, so the DNA of R-strain. So the DNA of one type of bacteria is added with the another one type of bacteria. Another one type of bacteria means just imagine S-strain bacteria. Okay, so the DNA of R-strain bacteria is added with the S-strain bacteria. This bacterial cell retains some important properties of the R-strain. Okay, the S-strain bacteria retains some important properties of the R-strain bacteria. This process is called transformation. Hope you understood, my dear students. So this is one of the important 5-mark questions. Okay, search of genetic material. Many 1-mark questions, the years and the scientist name. Okay, so it's very important for 1 mark question. So dear students, today we have completed two important topics. The first topic is properties of gene and the second topic is search of genetic material. And we studied two more important 2 mark questions, one gene, one enzyme hypothesis, and one gene, one polypeptide chain hypothesis. So dear students, hope you all enjoyed today's class. Today's class is also very easy class. Okay, this properties of genes is a very easy and very small 5 mark question. So dear students, all study and try to complete these two question answers today itself. Okay, next we will meet in the next class. Thank you.